Hello, my dear students. Today, let us learn about the sex determination in plants and animals. How the sex of the offspring is determined, whether it is male or female, in what proportion male and female are produced, especially at the chromosomal level, how the sex male and female is determined. These aspects let us try to understand. Scientific studies on the sex determination started as early as 1891. Before that also some attempts were made, some hypotheses were made, some assumptions were made about the sex of the offspring, but they had no scientific basis. Hanking in 1891, while studying the spermatogenesis in insects, observed a nuclear body that determined the maleness. In male insects, he observed a special body, nuclear body, presence in the nucleus. He named it as the X body and said that X body determines the maleness. If the X body is absent, then it is female. Later investigators proved that X body of the Hankings is in fact X chromosome. At that time, the microscope was not developed, it was not sophisticated, resolution was not good. Even then, they tried to see the chromosomes, and people said that the X body of the Hanking is in fact X chromosome. McClung, an American zoologist, called the X body as Y chromosome, the same X body, but he named it as the Y chromosome, which determines the sex in the sperm of the grasshopper insect. Stevenson discovered that one of the chromosomes in the male mealworm beetle was heteromorphic. One chromosome in the heteromorphic pair was similar to chromosome in the females. This is called X chromosome. Male mealworm beetle was heteromorphic. One of the chromosomes in the pair, heteromorphic, they are different, either in the width or in the length or with respect to the centromere position. The two chromosomes were different, sex determining chromosomes. One chromosome in the heteromorphic pair was similar to the chromosome in females. One of the chromosome in the males was similar to the chromosome present in the female. That means one particular chromosome is shared by both the male and female. Wilson observed the same chromosome in other insects, other organisms. Morgan, T.H. Morgan, Thomas Hunt Morgan, studied the sex determination in Drosophila insects. He found that the female fly has 4 pairs of chromosomes. 4 pairs, 8 chromosomes, 4 plus 4. 3 pairs, autosomes, A for autosomes, plus XX. In females, 3 pairs of chromosomes are autosomes. XX chromosomes are allosomes or sex determining chromosomes. and male fly has 3 pairs and heteromorphic pair chromosomes XY. These are heteromorphic. X is different from Y. Y is different from X. Therefore, they are called XY or heteromorphic, morphologically different. Past views, even before the scientific investigations were made about the sex determination, even after that also, some views were expressed. They can be broadly divided into 5 types. Sex of the organism is predetermined before the fertilization, progamic. That means at a gametic level, before fertilization. One view. Second view, sex is determined at the time of fertilization, syngamy. Fertilization process. Which gamete fuses with which gamete of the male. This will determine the sex of the next generation. Third, sex is determined after the fertilization. One view. After fertilization, this is called epigamic, this is syngamic, this is progamic. Before fertilization, during fertilization, after fertilization. Nutritional and environmental factors. Some believed that the nutrition of the male or nutrition of the female will determine the sex of the offspring. Age and vigor of the parents. There are also some views, age of the male, age of the female, and also vigor will determine. These were the views, different views with regard to the determination of the sex. Now it is confirmed that sex is determined at progamic and syngamic. Cytologically, it is proved, it is confirmed. The sex is determined either at the gametic level, formation of the gametes, or at the fertilization level, progamic or syngamic. Then autosomes, allosomes. The chromosomes that determine the sex of the chromosome are known as allosomes or sex chromosomes. The chromosomes that determine the sex of the individual are known as allosomes or sex chromosomes. Other than allosomes, autosomes. Other than allosomes are many times in many individuals, many times more than the allosomes or sex chromosomes. Sex chromosomes will be generally a pair, but these will be more. Autosomes will be more. For example, in humans, 44 autosomes. In females, XX, 44 XX female, 44 XY male. Then 44 autosomes, XX or XY allosomes or sex chromosomes. This is the situation in human beings. In Drosophila, total 8 chromosomes. Out of 8, 6 are autosomes, 2 are allosomes, sex chromosomes. XX female, just like in humans, the femaleness is determined by XX in Drosophila also. In males, XY. So gametes pair, for example, here gametes, only one type of gametes will be produced, 3AX. But here two types of gametes will be produced, 3AX one type, 3AY another type, 50-50. But here 100% same, even in the human beings also. Sex determination in animals. right from the lower animals to higher animals, human beings, sex is determined, but in different ways. Sex determination chromosomes are different, alleles are different. Male and female differ with regard to the sex chromosomes. One method is XXXY, XX female, XY male, XXYY method. This method was first observed in the milkweed bug Ligaeus tursicus by Wilson and Steven, hence it is known as Ligaeus type. Look at this. XX female homogametic, both are similar, similar. XY male heterogametic type. Heterogametic type, because X is different from Y, Y is different from X. Morphologically, length or centromere position or the number of genes they bear are different. Therefore, the XY chromosomes are heteromorphic, whereas XX are homomorphic. This type of sex chromosomes are found in man, Drosophila, goats, mice, etc., most of the mammals. Then ZW female heterogametic, ZZ male homogametic. In man, male heterogametic, female homogametic. But here, female ZW, Z is different from W. But here in males, both are similar. This Z chromosome is shared by both male and female, but the female has a different chromosome W that determines the femaleness. This situation is found in butterflies, moths, birds, codiflies. Then X0 type, Y chromosome missing, female XX. When you compare with the human beings, look at here, female XX correct, but male XY, but here Y is missing. Example, Orthopteran and Hymenopteran insects. ZO type. Here, Z or Y chromosome is lost. ZZ male homogametic, ZO female, here missing, Z is missing, here Y is missing, male, Solinobia. Then multiple sex chromosomes. In this type, either X chromosome or Y chromosome will be represented by 2 or more chromosomes, which promote maleness or femaleness. For example, instead of XX, XXX. Instead of XY, XYY. This is multiple sex chromosomes. Sex is determined by an extra chromosome. So this is how the sex is determined in human beings. Male, female. Male produces, male sex chromosomes are XY, female XX. Males produce 2 types of gametes, one bearing 50% bearing X chromosome, 40% bearing Y chromosome. But here, female produce only one type of gametes. All those gametes carry X chromosome. So if you take different combinations, any gamete may combine, fuse with any type of gamete, So if you take into account all possible combinations, then XX, XX, XY, XY. XX is female, female, male, male. Therefore, in the offspring, statistically speaking, 50% male children, 50% female children. But this may not happen sometimes, because that depends upon which male gamete fuses with the which one. So statistically, genetically speaking, 50-50. So this is represented in chromosome wise. 44A autosomes, sex chromosomes, XX female. The male gametes bear 22 autosomes, X sexochromosomes, but here 50% 22A X, 22Y. Progeny female, male 50-50. Sex determination in Drosophila is also similar, because here also, just like human beings, female XX, male XY, therefore two types of gametes in males, only one type of gametes in females. If you take into account all possible combinations, male, female, 50-50 in the progeny. Then ZW, ZZ, butterfly. Butterfly, moth, codiflies, birds, all birds. Here, female produces two types of gametes, but in human beings, only one type of gametes. Remember, in human beings, female produce only one type of gametes, but here two types. But here, males produce only one type of gametes, reverse. So, females, here progeny, ZZ female, ZW, 50-50 again. XX, X0 Hymenoptera insects. In these 20 autosomes, XX female, X0 male. Then here, only one type of gametes. Here, two types of gametes. One carrying the X, another carrying the 0. So, females XX like this, males here X0. This is in Orthopterans, Hymenopteran insects. ZO type Solenobia. In this, male ZZ, female Z0. Here, two males produce two types of gametes, females produce two types. But here, males produce gametes, but all of them carry the Z. Here, one type 50% carry the Z, 50% carry the O. Male ZZ, female ZO. Then haplodiploidy sex determination in honeybees. Honeybees colony, they are social insects. Altogether, different types of insects live as a colony. They are social insects. Chromosome number, female 32, male only 16. Important, remember. In honeybees, females diploid 32, males only 16, half of the chromosomes. There will be three types of individuals in a colony. One is called queen, that is diploid, carries 32 chromosomes. It develops from fertilized eggs. It develops from fertilized eggs. Queen diploid, 32 chromosomes. It develops completely, very size-wise big. It takes more nutrients. Fertile. This is fertile. Queen is fertile. Well developed. Diploid. Diploid workers develop from the fertilized eggs, underdeveloped, non-fertile. Just like a queen, they are also diploid, 32, but underdeveloped, undernourished. So therefore they are non-fertile. They work as the workers. Then haploid males, also known as haploid drones, they are haploid. Males are haploid. They are fertile. They can produce the sperms. Even though they are haploid, they produce the sperms. You can ask how. Here, during spermatogenesis, mitotic divisions take place, not meiotic. But here, when it produces gametes, meiosis division takes place. So 32 will be reduced to half, 16 chromosomes. But here 16 remains. Sexually functional, produce spermatozoa by mitosis. So this is how haploid queens fertile. Workers, diploid workers are not fertile. It produces gametes, meiosis division, 16 chromosomes. But here sperms by mitosis. So fertilization takes place. N plus N, 16 plus 16, 32. Diploid female. So these, in the diploid females, out of many, only one will develop into queen. It will be nourished with high nutrients, rich nutrients, grows well, remains fertile. But these are undernourished, they remain sterile. They do not reproduce. Then here, unfertilized. The female produces gametes. These are unfertilized. Unfertilized, so therefore 16 chromosomes. These develop into males, male drones. How? Without fertilization, by a process known as parthenogenesis. Remember, parthenogenesis. Haploid males N16. Remember, diploid queen 2N fertile. Then diploid workers female. They are also diploid but non-fertile. Then males are haploid. Males are haploid, developed by parthenogenesis process. So this is a gynandromorphs, half male, half female. Because of the abnormal mitotic divisions, this X chromosomes will be lost. Half of the body will be male, half of the body will be female. Bilateral gynandromorph Drosophila. Just our mythological Lord Shiva. Then this is another interesting hormonal control of sex, Bonelia. The sex is determined by hormones. This is a female. very large when compared to male. Male will be very small. If this male enters into this, it becomes a male. Suppose undifferentiated one, if enters and settles here, it becomes male. Suppose some of the eggs, they do not lodge here, just like a stigma or proboscis, they free living, they become female. Some of them settle down here and they become intersex. Here, male very small grows inside the body of the female. In other words, the female hormones determine that it should be male. Those which are free living, eggs, larvae or other things, they become free, they become females, and afterwards they grow like this. Some of them grow here, they become intersex. Sex determination in plants. So far we have studied the sex determination in animals, but what is the case in plants? In plants also there is a lot of variation. Not all are similar, all flowering plants are not similar. The sex determination has been very well studied in dioecious plants, where the male and female flowers are produced on different plants. Male plant, female plant, male plant produces the male flowers, female plant. In such type of plants, sex determination has been studied because it is easy. Then here, just like Humulus, Melandrium, Vallisneria, Dioscorea, Pagaria, Rumex, these are no sex chromosomes. So this is a situation in plants. XX, XY, XX, XO, XX, XY, male heterogametic, female with an extra chromosome. Here XY female, XX male. Here XX female, XY, Y chromosome extra. This is in Rumex, Humulus, Japonicus. So this is how we understand the sex determination. Sex is determined progamic or syngamic situations. And in different animals, different plants, the sex is determined in different ways. Sex chromosomes or allosomes, other than sex chromosomes, are called allosomes. Thank you.